Hello everyone, we are Group 8 presenting our project, The Greenhouse Trap, the combined impact of temperature and nutrients on algal blooms. As global warming drives up to tropical lake temperatures, it combines with agricultural fertilizer runoff to trigger toxic algal blooms, a devastating process known as eutrophication. The primary objective of our research was to stimulate this greenhouse acceleration in a controlled environment and isolate how heat and fertilizer interact to cause rapid ecological collapse. Here is it, our methodology speaker. Now we go to the problem statement. The purpose of this investigation is to evaluate the individual and combined effects of elevated water temperature and mimicking agricultural nutrient runoff fertilizer on the acceleration of algae growth in freshwater ecosystem. By monitoring variables such as turbidity, surface coverage, and coloration over time, this experiment aims to simulate the real-world environmental problem of eutrophication. Ultimately, the study seeks to determine whether the effect of both high temperatures and nutrient pollution creates a significantly bigger problem than either factor operating independently.
So, our aim of this study was to find out how temperature and nutrients affect algae growth in freshwater. We wanted to see whether higher temperature increases algae growth, whether fertilizer promotes algal blooms, and whether these two factors have a stronger effect when combined. By doing this experiment, we were able to better understand how climate change and nutrient pollution can contribute to algal bloom problems in freshwater ecosystem.
Okay, next is experimental setup. Okay, for the aim is to investigate the effect of nutrient input, which is fertilizer, and increase the temperature of algae growth. Okay, for the apparatus of the experiment, for transparent plastic bottle, 1.5 liter, lake water, commercial fertilizer, heat lamp, digital thermometer, marker labels, and smartphone camera. Okay, for the bottle arrangement, we have 4 bottles total. We have bottle A, bottle B, and bottle C and D. Okay, so for bottle A, it consists of no fertilizer and no heat lamp because this is a control. Okay, for bottle B, we have fertilizer and we have no heat applied because this is to show that nutrient enrichment only. For bottle C, there is no fertilizer but there is a heat lamp to show that temperature increase. And for bottle D, we have both fertilizer and heat applied to show that nutrient enrichment and temperature increase. Okay, for the expected observation from this experiment is bottle A will show minimal algal growth, bottle B will show moderate algae growth, bottle C mod- also show moderate algae growth, and bottle D is highest algal growth that show eutrophication effect. Okay, that's all.
Now for the methodology parts, to guarantee a perfectly controlled baseline, we first hooked small ventilation holes at the top of 4 upcycled plastic bottles to allow natural gas exchange. We filled each bottle to exactly 3/4 capacity with tap water and inoculated them with 2 tablespoons of alga-rich lake water as a uniform starter culture. We introduced 1 teaspoon of commercial fertilizer to bottles B and D, leaving A and C as controls. We activate our heat lamp daily from 9 a.m. to 5 p.m. over a 4-day timeline, taking digital thermometer readings at 5 p.m. to verify our simulated tropical targets of 30 to 35 degrees Celsius. Now moving to our results. First day is 18 May, as you can see here. And then the second day is 19 May. The third day is on 21 May and the last day is on 22 May. Moving to our results, look at our 4-day growth curve. While the control bottle A remained completely clear, our individual state-ups show moderate shift. However, bottle D, which combined both the heat lamp and fertilizer, shot up exponentially to a critical score 4, turning dark green and highly turbid by day 4. Critically, our daily logs show that water temperature was not a constant variable. It actively climbed to 32.7 degrees Celsius in bottle D. This reveals a dangerous, self-reinforcing climate feedback loop. The fertilizer fueled rapid algae multiplication, which darkened the water column, trapped more radiation from our lamp, and spiked the temperature even higher. As shown in our discussion, this experiment perfectly traces a deadly chain reaction. When a climate-driven algal bloom reaches its peak and dies, a massive bacterial decomposition phase takes over. These bacteria consume all dissolved oxygen, dropping the water body into a state of anoxia, creating a toxic dead zone marked by mass fish die-off and foul odors. Our heat lamp simulation directly proved that the greenhouse effect acts as a multiplier for freshwater destruction. To break this chain reaction, we present three future forward recommendations. Impact and public message. For the impact of eutrophication on the ecosystem is very big. The algal bloom and the rise in temperature will affect the marine life in the freshwater ecosystem, as the water body has lesser dissolved oxygen and they will go into hypoxia due to the lack of oxygen, causing our food security to be a problem and our daily needs to be in the rates. For the public message, global warming contributes to many factors in our ecosystem. It doesn't only harm us directly, but also indirectly, water and food. We, as mindful civilians, must do our part by being mindful of our daily lifestyle or work that contributes to this phenomenon. First, ultrasonic bursts use targeted sound waves to shatter the algae's internal air pockets, forcing them to sink to the dark lake bed. Second, floating solar panels create large shading zones to block direct sunlight. Finally, modified bentonite clay binds to dissolved phosphates for permanently burying the algae's food source into the lake floor. By starving the algae of both surface light and nutrient fuel, we can shield our vital tropical water systems. That's all from us. Thank you and have a nice day.